welcome very much here. I'm very sorry but we don't have a pointer so far, so let's see if we get one. So I will start from here from the table. So the title of my talk is Visual Ecology of a Three-Level Parental Prey System. And it's work in collaboration with, as you can see, there are several researchers, all of us from Uppsala University in Sweden. So, as a short introduction, I would like to mention why has color vision evolved in animals. There might be three main reasons. We don't know which one it was the first one. Probably food localization, but they are also used in interspecific communication and predator avoidance. And we have a very nice example there with these monkeys, whose females are polymorphic for the photoreceptors in the eyes. Some females are dichromatic, other ones are trichromatic, and as you can see on this Mickey Mouse picture, you can see how they might experience the surroundings. And because this has, color vision has evolved, we have an extraordinary diversity of color signals in nature, and all of these examples you can see there are mostly related with sexual selection. So you can imagine that one of the advantages of having conspicuous coloration might be that you get more mates. And I'm not going to talk about aposematic coloration in this talk, but some of these advantages can be that you are more predated, or you can eat less if the prey detects you more easily because you are more conspicuous. And there, luckily for us, conspicuousness is subjective, so depending on the photoreceptors you have, and also in the light conditions and the color of the background, you can experience environments in a different way. This is a very classic example of how we perceive the color of a flower as humans. We are trichromatic, so we have blue, green, and red photoreceptor cells, and we see this flower in yellow, while bees, for example, have UV, blue, and green, and they see the flower in a completely different way. This is very interesting because there is opportunity for the evolution of private communication channels. This is a very good example for this. There are two or three examples only. This is the northern sword-tailed fish. And you have here the male and the female. And the male displays this coloration which reflects UV. The predator, the Mexican tetra, cannot perceive the UV because it doesn't have UV photoreceptor cell. So this male is displaying in a safe environment, okay? But we don't know what's going on with the prey, for example. So this is my study system. We have a damselfly with very conspicuous wing coloration. This wing coloration is sexually selective. It has a proven cost in predation by birds and potential cost in prey capture. In our system, the damselfly has 3-4 photoreceptor cells. We don't know so far. 3 as most of the insects, probably a 4th one in red. It's predated by this passerine bird, the white wagtail, in northern Scandinavia, in Europe. This wagtail has UV, blue, green, and red photoreceptors, and at the same time the damselfly predates on small dipterans, and we use as a model the fruit fly, which has 2 UV photoreceptors, blue and green. We ask a couple of questions here. Is there a private communication channel for the damselfly going on, in both ways, or just in one? And which are the costs of being so conspicuous? And to make things a bit more complicated, we included a second species of damselfly. They can co-occur in Europe, and when they co-occur, there's potential for reproductive character displacement. It's been shown that the size of the coloration, especially in the males, can change when they co-occur in sympatry. And as you can see, they are sexually dichromatic. This is the male in green, female, male and female. And what did actually do? Well, we studied how wing coloration is perceived by each of the visual systems on a patchy background. That means that if we have a damselfly, as you can see here, and this damselfly perches on a natural background, as this river, which is the opportunity for the visual system to distinguish, to tell apart the coloration of the wing from this patchy environmental background. So for that we use noise, receptor noise modeling and a discriminability index. And for that we require 4 main pieces of information. The first one is the eye sensitivity peaks for each visual system. For the bird, we know that because we can use the one from the great blue tit, from the great tit. which is very similar to the white wagtail. For the flies, we know it as well, but we didn't know that for the damselfly, so we did electrophysiology in the damselfly eyes. We also need reflectance spectra of each damselfly, so we did a lot of fieldwork. We sampled in total 18 different populations, at least around 20 males and 20 females per population, in sympatry and allopatry. And then we studied the wing reflectance using spectrophotometry. We use tele-spectrophotometry for estimating how these backgrounds look in terms of reflectance. And we also use standardized conditions for the model. Okay, so here you have some results. For electrophysiology, we found here in the x-axis you see the wavelength, and here you see the response in millivolts. So this is how much the eye is stimulated by our pulses of light of different wavelengths. And we found 3 very well supported peaks in UV, blue, and green, as most of the insects, so this is not so surprising. And a 4th potential peak in red, which was not very well supported by our data because a lot is going on here. We think that it might be because the photoreceptor cell is located very deep in the eye. And you can see here the results in spectrophotometry in terms of wing reflectance here in the y-axis along the different wavelengths. Males on top, females at the bottom. You can see that males have 2 different peaks. So Virgo, Calypteris Virgo males are more greenish, the other ones are more bluish, and they have different amount of percentage of reflectance. And the females, as you can see, they are pretty similar between each other, but Virgo is more brownish, so that's why this peak goes up here, while the other one is more greenish, that's why we have this peak here. And very important for you to know now is that if you look at the dash and the solid lines, they reflect allopatric and sympatric populations. There is not a big difference between them. Okay? So we move on, and we use all this data to estimate this discriminability index, which you have here, but I'm not going into details now. Is this y-axis here, the D, and so the larger the D is, it can tell apart more easily the coloration of the damselfly from the background. Values below 0 means that they are cryptic colors if you compare them to the background. So we have here how the damselflies are seen by the birds, by the same damselflies, and by the fruit flies. If we inspect first the males on top, you have different colors of box plots. The blue is for splendens, the yellow is for Virgo damselflies, and you have also allopatry and sympatry. So if you look at the males first, we can see that birds are doing much better at discriminating the damselfly coloration from the background. Then the damselflies do kind of okay, and then the flies do very poorly. There are no significant differences between allopatry or sympatry, so we can discard this potential character displacement. Now if we move on into females, we see here a very interesting pattern. Most of the females are below 0. That means that most of them are cryptic in the eyes of the beholder, except for Calypteris Virgo females, which are perceived by the birds. Okay, so far so good. So we have no color displacement. The wing coloration seems to be confusing, according to this index. Females are very cryptic. Is there a private communication channel going on? We cannot tell much about females because most of them are cryptic. But we can tell that the males, at least, they have a private communication channel going on with the flies because the discriminability is higher in damselflies than in flies. But this is not occurring regarding the predator. And finally, a very interesting point here, if you look here, most of the females are supposed to be not distinguished by the young species. And this is very interesting because if you go to the field and males are territorial and usually react to females, but when the females are perching, males do not move at all. So it means that probably when they are perching, females are totally cryptic, and they need motion cues, which we have not quantified in this study, to distinguish them. Okay, so then we wanted to know if this index was reflected in nature. So we estimated predation risk in several natural populations. These white wagtails are very interesting because they have feeding stations, so you can go there and collect wings from the damselflies, and you can tell apart forward hind wings and right and left wings, depending on the relative components of the species in the population. Sorry. If we look here, we would expect that males are more predated than the females, right? And this is what we found. Males of splendens are 4 times more predated than the females, while in Virgo is 1.3 times more. If we look at the males, we should expect similar predation levels in the males and larger predation in Virgo than in splendens females, right? Okay, so this is what we found. In males, this is not supported. So Virgo is 4 times more predated than splendens, so there's something else going on here. But in females, this is very clear. So splendens was kind of cryptic, and actually Virgo was 25 times more predated than the other one. So since we couldn't explain very well this pattern with our results, we moved on into some behavioral choice experiments in the lab. So you have here an experimental arena where we showed different morphotypes of damselfly wings with different coloration on the wings depending on the species, or different amount of coloration. And we used great tips for inspecting a couple of questions here. So the first one, is there an inherent preference for spot color or size? That might explain this tendency, but there is no. There is not. And then we also asked if there's a learned preference in the spot size. Because the white wattles arrived in bred from Southern Europe, and they arrived in Sweden before the damselflies emerged. So they might, and one of the damselflies, Virgo, the more predated one, is emerged before than the other one every single year. So it might be that they learned to recognize one of the morphs. And we found that maybe because there was very well support for gray ticks, but when we redid the experiment again with blue ticks, these guys didn't distinguish at all from the learned and the non-learned preferences. Okay, and so for finishing, this is what we are doing right now. We are going to do more behavioral experiments in the autumn or spring next year. And now we are running, we did this 2 days ago. We are marking, this is Nura damselflies in the field. We have controls with a number, and we have experimental damselflies with a number and some coloration on the wing. And we want to know if there is a weight increase or decrease in the natural population after some days, because they cannot access so well to the prey. And we also are going to do some experiments in the field, expecting the hunting success on the Drosophila damselflies. Flies.